synthesis. Yeah. So what happened is a lot of biological things that will happen here. Yeah? So it's totally different. Yeah? So so the synthesis of peptide and protein can be done chemically. Yeah, you can synthesize all these amino acids, the reagents, coupling agent, cleavage reagents, but also it can be done biologically. You can ask cells, either bacterial cells or human cells, to produce the protein or peptide that you want. Okay, the reaction is a bit different because cells have a more complex machinery. Yeah, but the end product is the same. Yeah, the end product is polypeptide. Yeah. So today we'll see how to synthesize protein biologically. Yeah. So we'll see what nucleic acid is, DNA, RNA synthesis, protein synthesis, and then there is a control mechanism as well for the protein synthesis to make sure that the protein is of a high quality. Yeah. And protein degradation. So what factors that will affect the structure of the protein after the synthesis. Right. So when we talk about protein, yeah, especially protein biosynthesis, we have to talk about nucleic acid as well. Yeah. There is a separate topic about nucleic acid, which we'll discuss it after we finish this. Yeah. And because this is the building block, yeah, this is the precursor of protein. Yeah, when we discuss about peptide chemical synthesis, we don't talk about nucleic acid because the precursor is amino acids. Yeah, but when we talk about the protein biosynthesis, then we have to talk about nucleic acids because cells will use nucleic acid to coat, yeah, to coat what protein that they can synthesize. Yeah. All right. So nucleic acid is made of chain of nucleotide. Yeah. So it's a polymer of nucleotide. So a nucleotide, yeah, consists of a base. So there are three parts, as you can see here. I will highlight. Yeah. So it has base. It's a nitrogenous base. Yeah. So it has a sugar. It has a sugar group. Yeah. Either ribose or deoxyribose, depending whether you have DNA or RNA, and phosphate. So there are three parts here. Yeah. So you have base, you have sugar, and you have phosphate group. Okay. So depending on the what kind of sugar that you have here in the middle, so you have either DNA or RNA. Yeah, either deoxyribonucleic acid or ribonucleic acid. So if you have RNA, so this is the kind of sugar you have. You have ribose. So when you have DNA, so you have deoxyribose. Yeah, so the sugar is different. The difference is here. Sorry, the difference is here. This is ribose. You see this OH, right? And deoxyribose, you lose one oxygen. So that's why it's deoxyribose. Yeah, you lose one oxygen. That's the difference. Even though you lose only one oxygen, but they are totally different. Yeah, DNA and RNA are totally different. Yeah. In human, we use DNA as our genetic code. So we inherit DNA from our parents, and we will pass this DNA to our offspring, yeah, to our children. Yeah, so our children will inherit our DNA. Yeah. And DNA has certain composition so that you inherit the character, you inherit the, all those qualities from your parents, and whatever you have now will pass to your children, and of course it will combine with your spouse's DNA as well. Yeah. So your children will not inherit you 100%, but of course will inherit you in 50-50, from you and your spouse as well. Yeah. So that's why your children is a bit different from you. There is no such thing that you are the same with your parents. No, it's different. Yeah. Or you are the same with your father, or you are the same with your mother. It's you are mixture, yeah, from your parents. Yeah, because each of your parent will contribute their own DNA. Yeah? So your combination basically. Yeah? So the more generation, the more distinct the differences are. Yeah. So because it's a mixture, you know, you marry someone from different races, you have your own DNA, your spouse has different DNA, you mix, and then your children will have a different character from your parents or grandparents. You know, you marry someone from other countries, you know, Caucasian or African, then your children will have a different characteristics as well. Yeah. So everything depends on DNA. Yeah? Everybody has different DNA. You know? No person has similar DNA unless you are identical twin. Yeah. If you are identical twins, then there is chance that you have a same DNA. Yeah? Even though still there are some differences. It can change. Yeah. So when you have a child, yeah, your child will have in terms of the DNA will have some differences. Yeah. Your DNA consists of around hundreds of billion of nucleotides, yeah, and your children will have around 200, yeah, 200 different nucleotides compared to you, yeah. It's not much, yeah, because you have billion, you know, you have a billion of nucleotides in your DNA, and your children have 20 out of billion of DNA, 20 nucleotide different compared, you know, out of billion of DNA that you have, yeah. But when you have your offspring, you know, the longer the generation, the differences will become more distinct. Yeah. So that's why when you compare you and your great grandfather, then the difference is a lot. You know. But when you compare you with your parent or you and your children, the difference is not much because the difference is only 200 nucleotides, billion of nucleotides in your DNA. Okay. So yeah, you use DNA as your code, you know, to come up with offspring that have similar character with you. But some organism like virus, they use RNA instead of DNA. Yeah. Just like COVID-19 virus, they use RNA. Yeah. They don't use DNA. They don't have DNA anyway. So they use RNA. Yeah. All right. So again, the structure, the nucleotide structure has three parts. One is the base, yeah, and then a sugar and a phosphate group, yeah, right. So just like protein, yeah, nucleic acids have a primary and secondary structure. As long as you have polymer, it has structure, yeah. As long as you have a long structure, you have a polymer. So this is a polymer of nucleotide, so it has structure. So for DNA, it is a double-stranded helix, yeah, and also it has a hydrogen bond between strands, just like protein, yeah? Just like protein. And for RNA, you have three kinds of RNA, yeah, we'll discuss today: mRNA, tRNA, and rRNA, yeah, all single strand, yeah. For RNA, all single strand, and it has a hydrogen bond within strand. Yeah? So this is an example of RNA, yeah, single strand. Yeah, a single strand. It's just create a unique structure so that there is a hydrogen bond between strand. Yeah, so it's single strand, right? So this is DNA. Yeah, so it's double strand, and it has a hydrogen bond between strands. Okay. So for DNA, because it creates a double strand, so there is a interaction between base. Yeah, there is an interaction between base. Yeah. So depending on what kind of base that you have, yeah, and the base between DNA and RNA are different. Yeah. For DNA, the bases that they have is ATGC, yeah, adenine, thymine, guanine, and cytosine. Yeah, this is the bases that belong to DNA. And for RNA, they don't have thymine, yeah? They have uracil instead, right? So that's the difference. So for DNA, what you have is ATGC. So for RNA, what you have is AUGC, yeah? So that's different, yeah? So, and the pairing is G will always pair with C, yeah? Guanine will pair with cytosine, yeah? And T will pair with A, yeah? So you have a G and C here, all right? So G with C, all right? And then T with A, yeah? So G and C is connected, is interacting through 3 hydrogen bond, yeah? And T and A is interacting, they are interacting with 2 hydrogen bonds, all right? So because of this, G cannot interact with A, T cannot interact with C, okay? So that's what happened, yeah? Well, but sometimes, even though G will always pair with C, T will always pair with A, sometimes in our genetic code, there are some changes due to mutation, yeah? So nucleotide change by itself, yeah? And this base pairing is disturbed, right? So it disturbed. So it, in some cases, it causes cancer, yeah? But fortunately, in our cells, we have that mechanism. We have certain mechanism to correct that mistake. So you have to keep in mind that that the genetic code that we have in our body, the DNA, yeah, it will always replicate, yeah. So there is a copy paste a process that will copy the DNA, yeah. So we don't lose our DNA. It will copy DNA, yeah, copy DNA. And during this process, there are always mistakes, yeah. It should be G, suddenly it becomes T. It should be C, suddenly it becomes T. It should be A, suddenly it becomes G, yeah. Because billion of nucleotides, as I mentioned, yeah, billion of nucleotides, and your cells have to copy, have to replicate, all right? And sometimes mistakes happen. When that happens, then the base pairing will be different, yeah? It will be different. So in some cases, as I mentioned, it will cause cancer, yeah? But magically, you have a correction mechanism, yeah? If for some reason G becomes A, you know, because of mistake during the replication, during the copy paste process, then your cells will change it back to G. Yeah? When it should be C, suddenly it becomes T, then your cells will change it back to C. Yeah, because if we don't have that correction mechanism, then we will easily get cancer. Okay. All right. So basically, this process happen all the time. Yeah. So it's happening now in your body. Yeah. The copy, the replication process of DNA. Yeah. And the correction mechanism always happen as well. Yeah. I have that slide, if I'm not mistaken, not in this slide. Yeah. So in different slides, I explain to you how our body correct, how our cells correct that mistake during the replication process. Right. So DNA is the genetic material, yeah. So it's located in 23 pairs of chromosomes. So you have chromosomes, yeah. This one, yeah. So you have 23 pairs, and that's your blueprint, yeah. In these 23 pairs of chromosomes, that's your blueprints that you know that you inherit from your parents, right? So actually it's long, you know, it's a long chromatin. Yeah, this is a chromatin. Yeah, so it's long. Yeah, if you extend, it's quite long. Yeah, it's just to maximize the, you know, to maximize the space. So that's why they fold. Yeah? They fold and they pack into this kind of structure. Yeah, like egg structure. Yeah. So you have 23 pairs, yeah. So each pair is different. Each pair has a certain DNA with certain function, yeah. So it's chromosome number 1, 2, 3, 4 until 23, and each chromosome has a specific DNA for specific function, yeah. For example, the DNA that controls the color of your hair is located and it is located at chromosome, it's certain chromosome, yeah. So the DNA that controls the color of your skin is located at certain chromosome as well. The DNA that controls your you know, your intellectual, yeah, your how smart you are, is located in certain chromosome as well. Yeah. So each chromosome is different. Yeah. So each chromosome is different. So DNA has two functions. Yeah. It will replicate to reproduce itself when cells divide, because your cells always divide unless you die. Then you die, your cells stop dividing. Yeah? As long as you're still alive, then your cells will divide, because your cell have to replace. You know, when your cells died, it has to be replaced, right? So during this, when new cells replace the old cells, then your cells have to divide. Yeah, and during this process, replication process happen to make sure that the new cell will have exactly the same information as the old cells. Okay, we don't want that the new cells have different information than the old cells. Then you become mutant. You know, you become suddenly overnight you become a new person. We don't want that to happen. Yeah, but sometimes it happens during aging process. Yeah, uh, like 10 years ago I don't have gray hair, now I have gray hair, you know. So that's mutation process. Yeah, because because the new cells are different from the old cells, mutation happen, I got gray hair, you know. My skin gets more wrinkle, you know, when we get older. So during the aging process, so the cell division, yeah, will produce a new type of cells. All right, that's different from the old cells. So that's why, you know, when you get old, you look different. Okay. And also, DNA will transmit the information, yeah, from during protein synthesis. So to make sure that you become an individual that will inherit the character from your parents, yeah. And to make sure, as I mentioned, to make sure that when the cell divides, the new cell will have the same information as the old cell. Okay. All right. So DNA contains genetic information. So as I mentioned to you, that the DNA has nucleotides. Yeah. So the nucleotides is your genetic information. It's like a binary code. Yeah. It's like binary code 01010101. Yeah. It looks simple, only 0 and 1, but in your computer screen, you see something beautiful. Yeah. This a very simple binary code is translated into graphics. Yeah. You can play games in your computer, you can watch movie in your computer, you can listen to my lecture this morning if you're not sleeping. Yeah. If you are sleeping, I don't know. So all those things that you are seeing, they come from binary code. Yeah, they come from binary code, from 0 and 1, 0, 1, 0, 1, 0, 1, right? So the similar thing with genetic material. Yeah, all that we have in our body, they come from 4 letters. Yeah, from U, A, G, and C. All right. So that's your genetic information. That's your binary code. Okay. So they code for particular protein depending on what kind of nucleotide base that you have. Yeah. I'm sure you learned this in your high school. Yeah. When you have UUU. Yeah. So you have phenylalanine, yeah. When you have UUA, you have leucine, yeah. When you have CUC, you have leucine, you know. So leucine can be coded by 2 combination, yeah, or more, yeah, at least by 2 combination, yeah. When you have proline, when you have CCU, CCC, CCA, or CCG, then you have proline. All right. So this is your binary code, yeah. But it's not binary. It's not 0 and 1. It's 3. Yeah, it's 3 nucleotide. Okay. 3 nucleotide will give you 1 amino acid. So this is what you have in your DNA. You know, you have a very long DNA. You know, and you have 3 nucleotide. So each nucleotide, each 3 nucleotide will give you 1 amino acid. All right, so so that's DNA and RNA. Yeah, the function is different. Yeah. There are 4 RNA. Yeah, one is mRNA, it's messenger RNA. So this RNA carries genetic information from DNA in nucleus to cytoplasm, yeah, where protein is synthesized. So the protein is synthesized in a compartment. It's called cytoplasm. Yeah. So it's a plasm. Yeah, it's a plasma inside the cell. So that's why it's called cytoplasma. Cyto is cells. Yeah. So that's messenger RNA. Yeah. So in certain organisms that they don't have DNA, they have mRNA. Yeah. So they don't need DNA. For example, COVID-19 virus. COVID-19 virus doesn't have DNA, but they have RNA. And the type of RNA they have is messenger RNA. And there is a transfer RNA as well. Yeah. So for transfer RNA, the job is different. Yeah. tRNA carries amino acid from amino acid pool to mRNA, so it will bring the amino acid. Yeah. From amino acid pool, so there is a store. Yeah. There is a place where amino acid is kept. Amino acid is stored. Yeah. And transfer RNA will bring each amino acid to mRNA, depending on the sequence of mRNA. And then another type of RNA is called ribosomal RNA or rRNA. Yeah. It joins with ribosomal proteins in ribosome. So ribosome is is the machine that synthesizes the protein. Yeah. Inside the cytoplasm is a machine. It's called ribosome. It will join the ribosomal protein where amino acid join to form protein primary structure. Yeah. So I'm sure you still remember from my introductory slides. Yeah? In my introductory slides, I show you how this thing happened. Yeah. So that's the function of ribosomal RNA. And then there is a snRNA, small nuclear RNA, is associated in protein in nucleus to form small nuclear ribonucleoprotein particles, yeah, which delete introns from pRNA. So introns is the a nucleotide that doesn't code any protein. Yeah. So as I showed you before, all these nucleotides that produce amino acid. Yeah. But for intron, it's part of the nucleotide that doesn't produce any amino acid or protein. Yeah. So it will be deleted during the process. Yeah. Right. So this is the information flow. Yeah. During the protein synthesis. Yeah. So all the information is stored in DNA. Yeah. So it's your binary code. Yeah. If you imagine computer, it's your binary code. So it's stored in DNA, and then transfer to RNA, and then from RNA then expressed in the structure of protein. So DNA to RNA, and then to protein. Yeah. DNA, RNA, protein. DNA, RNA, protein. Yeah. This is dogma in biology. You have to follow this. Yeah. You cannot run away from this principle. So that's why it's called dogma. Yeah. This is the dogma in biology. Yeah. Yeah, from DNA transfer to RNA, then expressed in as protein. Yeah. There are two steps in this process. One is called transcription. So the information you have in DNA is transcribed into mRNA. Yeah, again, this is called transcription. And then translation. Yeah, you translate the information from RNA into protein. Yeah, so transcription and then translation. Okay. All right, so let's look at transcription. So this is what happened during transcription. Yeah, so you have DNA. Yeah, so the information transcribed from DNA into mRNA. Yeah, all this thing. I'm not going to explain to detail because this is too biology. Yeah, yeah. But basically, when you have DNA strand, yeah, you have nucleotide, yeah, you have nucleotide sequence. Then the sequence will be transcribed into mRNA, yeah, into mRNA, and with the help of an enzyme called RNA polymerase, yeah. So the function of RNA polymerase is to print the code. It's a printer, yeah, it's a printer to print the RNA, yeah, to translate from DNA to RNA. And then they print the sequence, yeah. So it starts from DNA helix unwinds, yeah. And then RNA polymerase will print the RNA based on the DNA, yeah. When the process finishes, then mRNA will move to ribosome. Yeah, it enters the ribosome to produce the protein, and the DNA will rewind. Yeah, so DNA opens, and then the RNA polymerase will type the RNA code. Yeah, will print the RNA code based on the DNA sequence. When the process happens, the RNA goes to ribosome, and DNA will close, will rewind. Yeah, will go back to the initial structure. This process is to control, yeah, to make sure that there is no error happens, yeah, because during the printing, when the RNA polymerase prints all the genetic code, yeah, sometimes error happens, yeah, sometimes mistakes happen, right? Especially when the DNA is long, so some mistakes will happen, yeah. So there must be some transcriptional control, yeah. And as I mentioned to you, you have some correction mechanism, yeah. So during that process, the mistakes will be quickly corrected. Okay. There are some organisms that don't have this kind of correction mechanism. For example, is influenza virus. Yeah. Influenza virus, they don't have this mechanism. So during the printing of their RNA, okay, when there is a mistake, yeah, when the RNA polymerase makes a mistake, instead of writing G, the RNA polymerase writes C, for example. This is not corrected. This is not corrected by the cells. Yeah. So as a result, influenza virus always mutates. Influenza virus always mutates. So that's why you get flu all the time. Every year you get flu 4 times. You get flu 4 times every year. Yeah. Children, they got flu like 7 to 8 times a year. Yeah. But once you get older, you go to school, you go to university, you get less and less flu because your body already remember all the virus that infect you. Yeah. Because all these virus, they mutate. Yeah. Their genetic code always change because of this process. You know, the RNA polymerase during the transcription process, they make a lot of mistakes when they type the genetic code. Yeah? When they write the genetic code, and these mistakes are not corrected because they don't have that mechanism. They always mutate. Yeah? Mutation always happen. But in human, we have that mechanism, fortunately. So you don't become mutant overnight. Yeah. Otherwise, you become X-Men, you become mutant, then everybody will get shocked. Yeah. The good thing is we have that mechanism. For COVID virus, they have that mechanism as well. So that's why for COVID virus, the mutation is slower than influenza virus. In influenza virus, the mutation happens quickly, around 15 nucleotides per year. So 15 nucleotides change per year. Yeah. And so that's the reason why there is no vaccine for influenza. There is no effective vaccine for influenza because the virus always changes. You create one vaccine for this year, next year, the virus already changed. The mutation changed the virus, so your current vaccine will not work. So you have to come up with new vaccine. So that's why there is new flu vaccine every year because there is no because mutation always happen. The RNA polymerase of the RNA polymerase of the virus keep making a mistake. Yeah, when they write the genetic code, yeah, in the virus. So, but for COVID-19, yeah, maybe you listened to the news, you heard the news 2 days ago that there is a vaccine, yeah, vaccine candidate with effectiveness of 90%. Yeah, that's the interim result. Yeah, the initial results shows that the effectiveness is 90%. Yeah. So there is a better chance to get vaccine for COVID-19 because COVID-19 virus, we call it as SARS-CoV-2, even though it's mutated, it always undergo mutation, but the mutation is not as fast as flu virus. So there is a hope that the vaccine that we are creating now will be able to defend you from COVID-19 virus. Yeah, we hope so. We hope so. Yeah. We just have to control so that the virus doesn't keep mutating because even though the virus doesn't mutate too much, but if you let the virus to circulate, if you let the virus to transmit from one person to another person, you don't wear masks, you don't wash your hand, you keep coming to crowded place, so the virus will jump from one person to another person, from one host to another host. So it will help the virus to mutate. We don't want that to happen. We have to stop that. So that's why you don't go to university. You stay at home. Your family stay at home. You wear masks. You don't go into crowded space because you want to stop virus from mutating. Because every time the virus jump from one host to another host, they will mutate. Okay. So by stopping the virus, by slowing down the mutation process of this virus, hopefully by the time the vaccine is ready, the vaccine will be effective for COVID-19 virus. Otherwise, if you let the mutation happen by spreading the virus to other people, then we will be in trouble because the vaccine will not recognize the virus anymore. Yeah. So that's the good thing about COVID-19 virus. So basically, COVID-19 virus is similar to us. They have a mechanism that can control the RNA polymerase here. Because RNA polymerase is quite a sloppy enzyme. They always make mistake when they copy the genetic code, when they write the genetic code, especially when the genetic code is long. Yeah. All right. So that's one of the control mechanism during transcription process. Protein synthesis occurs in ribosome. Yeah, so this is a ribosome. So it happens in the ribosome, right? Here. So ribosome has 3 sites. Yeah, A site, B site, and E site. A site is the site where the process starts. Yeah. P site is the process where the amino acid attach to each other to create protein. E site is where the polypeptide exit the ribosome. So it enters from here. Yeah. And the protein will come out from here. All right. So mRNA masuk dari sini. It will enter through A. And then the amino acid, yeah, brought by tRNA will attach here. They will join together into polypeptide, and the polypeptide will exit from this door, yeah, from the E side. Right. So so that's the translation process. Yeah. Translation process is protein synthesis. Yeah. The previous process that I explained is transcription process, where DNA is transcripted into RNA. And then once you have RNA, then the next process is translation. Yeah. You change the RNA into protein. Yeah. So RNA translated into protein in 4 steps. Yeah. Activation, initiation, elongation, and termination. Yeah. So this is elongation. Yeah. So of tRNA binds to mRNA. Yeah, tRNA, you should remember, it brings the amino acid, all right? And then it binds to mRNA depending on the sequence, yeah, at the A sites at the ribosome. So it enters the ribosome through A site, yeah, through A door, pintu A, yeah. Each tRNA is specific for one amino acid only, yeah. So, but some amino acid coded by up to 6 codon, yeah. If you remember, maybe I need to show you in the previous slide, yeah. Yeah, like proline, yeah. Proline is coded by CCU, CCC, CCA, CCG. Yeah. So one amino acid can be coded by more than 1 codon, yeah. We call this 3 nucleotide as codon. All right, so amino acid at A site link to previous amino acid, yeah. So they join to become polypeptide, yeah. So ribosome moves along 1 codon and next the RNA binds to A site. Yeah. So the process happens like a sewing machine, you know. Kalau you all jahit, yeah, sewing machine, you see that process. So actually it's similar, yeah. So the ribosome actually dia akan jahit, yeah. Dia akan jahit the polypeptide, yeah, one by one amino acid at a time. So and the product is the polypeptide. Termination, yeah, final codon of on mRNA contains termination signal, yeah. It will give a signal to stop the process, yeah. And then releasing factors cleave the polypeptide chain from tRNA, and the RNA will go back to, you know, will go back to the initial place without carrying amino acid, yeah. And then mRNA released from ribosome and broken down into nucleotides when the process is finished. Yeah. So each amino acid activated by reacting with ATP. Yeah, ATP is adenosine triphosphate. Yeah. So tRNA is synthetase. Yeah, there is an enzyme that helps the synthesis. Attach the activated amino acid to one particular tRNA. Yeah. So the function of ATP is just like the coupling reagent in the chemical synthesis of peptide. It helps the connecting to amino acids. Yeah. Right. So this is the molecule of messenger RNA. Yeah. This is mRNA. Yeah. This is a codon. Yeah. 3 nucleotides. So maybe it's easier for you to see the visual. It was made in the nucleus by transcription from DNA molecule. Sorry. So this is ribosome. Yeah. It's a machine that produces protein. So this is a tRNA. It brings amino acid. Yeah, it arrives. It brings amino acid to the first 3 codon on mRNA. Yeah. So it has to create a pair, U with A with U, C with G. Yeah. G with C is 3 hydrogen bond. A with U is 2 hydrogen bond. Yeah. All right. So and then the next amino acid. Yeah. GGC. So the pair is CCG. Yeah. What happened when you change G, when you change this G into A? you will not get CCG here. When you change this G into A, what you have is UCG in your tRNA. So that's the reason why when you have one nucleotide change into something because of mutation, the effect is very detrimental. Because even one nucleotide change, all right, you will not get this amino acid. Because when you change this G into A, as a result, you will not get C here. You will not have CCG in your tRNA. What you have is UCG, which is different amino acid, right? So if different codon still give you the same amino acid, is fine. But if different codon will give you different amino acid, then we call it as mutation. And cancer happen that way. Yeah, cancer happen that way because of the mutation. There's only one or two nucleotides, change the protein, then the cell growth is different. You cannot control the cell growth, then you get cancer. Yeah, it happens because of small mutations sometimes, right? And then peptide bonds, yeah, is created between two amino acids. The peptide bond formation is catalyzed by peptidyltransferase. Yeah, so this is the enzyme that helps the formation of a peptide bond. Yeah. The energy for peptide bond comes from ATP. Yeah, adenosine triphosphate. This is the battery of your body. So your body has a battery. The battery comes from ATP. Yeah. So the peptide bond formation results in the shift of the peptide. Yeah, from P side to the A side. Okay. So again, the first tRNA molecule release the amino acid, moves into the cytoplasm. Yeah, dia balik, moves back into the cytoplasm. And then the ribosome moves along the mRNA to the next codon. Yeah. So this process is repetitive. Yeah. It will repeat the same process over and over again until you get the protein that you want. This is the third tRNA. Yeah. It brings the next amino acid into place. Again, the same process happens. Peptide bond joins the second and third amino acid. And then the ribosome will continue with the next codon, and next codon. So this continues until a stop codon is reached. Yeah, stop codon is the codon that will tell you, hey, stop the reaction. Okay, no more tRNA, no more new amino acid. Stop the reaction. Yeah, then the polypeptide is then complete. Okay. So and then messenger RNA translated on polyribosome. Yeah. This is how you get your messenger RNA in polyribosome. Right. This is you have ribosome, 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 ribosome. So you have plenty of machine in your factory. Yeah. When you produce protein, you have factory. You have plenty of machine. We call it as polyribosome. So if one machine is broken down, you still have another machine. Don't worry. Protein synthesis is control, yeah, including the rate of protein synthesis. Of course, you must have control. If the protein is not controlled, it's for cancer. The protein keeps being synthesized. There is no way to control the process, then it means cancer. Cancer actually is the cell growth, cell division, yeah, the production of protein, but there is no control mechanism. Yeah, you keep growing, the cell keeps growing. There is no control mechanism. There is no mechanism to tell them, hey, stop. No such that mechanism. Yeah. So that's cancer. We don't want that to happen. Yeah. We don't want in our life to keep growing. Grow without any control. We don't want that. Yeah. Including with money. You cannot just keep collecting money without controlling it. Otherwise, it will become cancerous. You will suffer because of that. So keep that in your mind, that keep collecting money for the rest of your life unlimited amount of money actually is not healthy. It's just like cancer, you know. The only thing that can grow without any control mechanism is cancer. All right. Yeah. Anything else, they must have some control mechanism, including how you carry on with your life. You know, at the end of the day, you have to have family. You know, you have to get some money for your family. You have to work and everything. Just keep that in mind, that everything has to be put under control, including what you want. You need to control it. How you manage your life. You have to control it, because the only thing that go uncontrollable is only cancer. Yeah, and it's not healthy. Yeah. So the rate of protein synthesis, it has to be controlled. Yeah. So so it's controlled by ribonuclease. Yeah. This is this process is called post synthesis. Yeah. It's a ribonuclease. Yeah. It's a degradative enzyme. Yeah. So basically, ribonuclease is will stop the translation process. Yeah. So that's why it's called degradative enzyme. Yeah. Will stop when the process is too fast. It will stop the reaction. Yeah. Other proteins will stabilize and destabilize mRNA against degradation by ribonuclease. So it's just like when you drive a car, you have brake, yeah, and you have accelerator. You have brake, accelerator, brake, accelerator. So you have this control mechanism during protein synthesis as well. Okay. In mitochondria, yeah. So this is the one of the organelle that helps the respiration in your cell. Yeah, they have their own DNA. Yeah, that's a unique thing about mitochondria. They have their own DNA, and they have their own protein synthesizing machinery. Yeah. So their genetic code is slightly different. Yeah. So they synthesize small number of proteins. Yeah, because this is a very important organelle. Yeah, very important component in your cells. It helps your cell to breathe. So they have their own machinery. They have their own life, basically. They synthesize their own protein. Yeah, and because they have their own DNA. So after the synthesis, there are protein degradation processes. Yeah. Sometimes this process happen. Yeah. Your protein will undergo some degradation. Yeah. For example, when you have ubiquitin proteasome, it will degrade your protein by attachment of ubiquitin units. Yeah, small unit that attach to your protein and degrade your protein. Yeah. Sometimes your protein can be degraded in lysosome. Yeah, because in lysosome, inside the cells, you have this enzyme, catepsin and proteinase. It will degrade the protein as well. Yeah. And then calcium activated proteinase, you have a lot of proteinase in your cells and in your blood. Yeah, it will degrade the protein. Yeah, this is part of control mechanism as well, to make sure that you don't have too many proteins at one time. Yeah, so you have a lot of proteinase. It will degrade the protein, right? So yes, you have a lot of proteinase in cytosol of your cell and also in your blood. Sorry. So it balance basically, yeah, between protein synthesis and degradation. Yeah, so you have this kind of a control mechanism, as I mentioned before. Yeah, that's all for today. I know it's a very biology. Yeah, you go to chemistry because you want to avoid biology, but unfortunately you see biology today. Yeah, any question?
Good. What will the topic that will come out in test one?
Sorry.
The topic for that coming in test one.
Normally I don't answer that question. Yeah. If student ask me whether this topic will be asked during the exam, I will not answer that question, because I teach you not for the exam. If you come to my class just for your exam, forget it. I teach you not for the exam. My exam will be open book. My exam will be open book, yeah, because I don't want you to memorize all these things, to remember all these things. You will get crazy. I will not tell you what will be asked during the exam. The exam will be open book, open internet. So instead of using your memory, you have to use your understanding to answer the question. Yeah.
Doctor, I mean which topics? Topic 1, topic 2, not specific.
That's another question that I will not answer. Everything that I teach you will be asked during the exam. Please remember, you are third year student, right? Third year or second year student?
Both.
Third year student, right? Oh, both second year and third year student. I advise you, when you take a course, especially protein chemistry, this is elective, right? You don't take this course just for the exam. Don't be exam oriented. This is university, not school. Yeah. You take this course to get some understanding about protein, about cells, about life, not for your exam. If you take this course just for your exam, at the end of the semester, after your final exam, you will forget all these things. All my effort will be wasted. Okay. So I know this is quite difficult to understand, because for the rest of your life, you learn something just for your exam, right? I know that. But if your life is dedicated just for your exam, you will suffer. Just for a piece of paper, just to get A, B, or C, you will suffer. Your life will not have meaning. All right. So again, as long as I teach, when you ask me which topic will be asked during the exam, I will not answer. As long as you come to my lecture, you get the lecture notes from me, then the question might come from that slides or might come from other sources as well. I teach CHM3701 last semester. The exam is open book, open internet. They can cheat if they want to, but majority still got C. Because what? Because they expect the question will come from the lecture notes. Actually, no. The question will come from anything. So that's why they get shocked, because they always expect that the question will come from the lecture notes. Lecture note is just a guidance to help you to understand. But you still have to read from your textbook, from Wikipedia, from other sources, you know. So you have to change your mindset, start from now, that the only source of knowledge is not your lecture notes. You have to read other sources as well. You are lucky that I don't give you assignment, but if you study in the US university, in American university, after each lecture, you will be given assignment, a lot of assignment. So you have to have textbooks, and all these assignments don't come from the lecture notes. Their lecture notes is very simple. Sometimes it looks so ugly, you know. It's handwritten. You know, it's handwritten, because they expect students to read textbook, and there is always assignment after every lecture. Luckily, I don't give you that, you know. But you see, the quality of American student is much higher than other countries. Because what? Because they read a lot. They don't ask their lecture. When I was there is no student ask their lecturer or ask their professor, professor, what kind of topic will be asked during the exam? No such thing of that. They never ask that question, because they know when they come to when they come to class, they try to understand. All right, and the question will not come from the lecture notes. Okay, so as long as I teach you during the class, and then the question will come from that topic. Yeah, because it's comprehensive, especially from your final exam. Your final exam will be comprehensive from the first topic until last topic. Yeah. All right, so that's all for today. Yeah, so I'll see you on Friday then. Thank you. Assalamualaikum.
Okay, thank you very much, Doctor.